Welcome everybody. So for those who haven't been here on Tuesday, I'm Tobias Hamp. I gave the lecture on Tuesday and will be giving this one because Burkhard Ross is currently on travel in Brazil. So my topic is the homology-based prediction of protein function. I gave you quite an extensive introduction last time. I will shortly recap it and what we have done in terms of homology-based prediction so far. So last week and on Tuesday I talked about DNA protein sequences. Just again a short question. So how many of you have already heard the Predict Protein 1 lecture? Oh yeah, okay. Okay, so 2 of you haven't? Okay. So in school you usually learn a top-down approach to biology, going from human organs, deliver to the cellular level, and sort of bottom-up approach from DNA to mRNA to proteins. And what is in the middle is usually left out, and the reason for this is that this is subject to research still. And this lecture and this whole lecture series is actually how to continue from the protein level, so from the sequences of the proteins. Yes, I showed you this slide. So this basically shows. If you go from this protein here, so this is a protein shortly after translation, and such a protein may, for example, then bind to other proteins and perform its function in the cell. One question to you, just to warm things up. So how do you get from the DNA level to the RNA and to the protein level? So what do you need for that?
Messenger RNA?
Yeah. So DNA is a sequence of 4 nucleotides, right? So in mRNA you exchange one nucleotide with the other. I believe diamond with uracil. Yeah. The question to you is what are the things that do this transcription or translation? So where do they come from?
Ribosome?
That's correct, yeah. What is a ribosome?
It's a protein?
Sorry?
Can I say it's a protein too?
I'm sorry?
A protein.
A protein?
Yeah.
Yeah, it's true. So several proteins. Yeah. So the apparatus that is necessary for this for the generation of the proteins are proteins themselves. Right? So that's a loop basically. Right. So this is what we would ideally want to have. If we had this for all the proteins, we would need this lecture. But so on this level of detail, we only know a very few processes, maybe a dozen. So we need something better or something else to really make progress in the field. And especially, we cannot go over structure entirely. So because we have not the experimental means or the computational means to really know the structure of all the proteins, for example, in human. And one thing you can do is you can try to predict function directly from sequence without going to the structure level. And for that, you first of all need to define what you actually want to predict. So how do you define protein function? And for that purpose, I've introduced the gene ontology last time. So this is a controlled vocabulary that where you have all sorts of terms. So should approach 100,000 terms by now that describe all the possible functions a protein can have. The gene ontology is organized on 3 levels. So it describes a protein in 3 categories. So first of all, what is it on the chemical level that the protein does? In this case, up here, it binds ADP. So those are the little yellow things that come and go. This is the so-called molecular function ontology. Then you define a protein with the biological processes it is involved in. In this case, it is the synthesis of ATP. ATP is basically the energy currency in the cell that is also the energy your muscles need in order to move. This is called the biological process ontology. And finally, we want to know where in the cell it all happens, and in this case, it is the F1 ATP synthase in the mitochondrium. So mitochondrium is a little organelle. All right. Any questions so far? Okay. So here's one actual example of the annotation of a protein in the gene ontology. This is the human hemoglobin subunit alpha. It is a very well-studied protein. You can see here the 3 different annotations in the ontologies. You have 3 subgraphs here, so they are disjoint. There are no edges between them. At the very root of the tree, so it's not a tree, it's not a graph. It's rather like a rooted graph. But anyway, so at the root you have biological process, for example, cellular component, molecular function. And as you go down the hierarchy, the terms become more and more specific. So for example, biological regulation here already, antioxidant activity, or general catalytic activity. And finally, until you reach the leaf terms, where you really have the final functions of the protein, the most specific descriptions of the function. Again, a short question: catalytic activity. What do you call a protein with catalytic activity? Okay, good. Right. So I have introduced last time a very simple approach to function prediction that works surprisingly well. It is based on the principle of homology. Homology basically means that as soon as you have some sort of speciation, you will find that the species that are newly created are very similar to the ones that already exist. In the end, creating something like that. So those are different proteins. So this is the same protein in different organisms. You see that the sequences are all very similar. And so our idea is that if you wanted to predict the functions of a so far not annotated sequence in a so far not annotated organism, you simply search through a database of proteins that are already annotated, and you transfer the functions of the most similar protein to the one protein that you want to predict. And again, so what is this? So I asked this question already. So this is called an sequence alignment, and you should really know what this is if you really want to continue this lecture, because this is basically what every other lecture will be based upon. Again, I introduced nearest neighbor principle. So if you search for a query in the database and you find similar proteins, there are different variants of predictions that you can do. You can only look at the one best hit. You can look at the k best hits, or you can also weight the k best hits, for example, by their similarity to the query protein. This is one actual implementation of this concept. So I introduced 3 different concepts which were all developed in the context of exercises of this lecture about 4 years ago. Again, this is one such concept. It first of all calculates the so-called quality score, which rates the overall quality of all the results that you got back from your query against the database of annotated proteins, and also rates each coterm then individually, and in the end outputs a prediction where each score is where each coterm has its own score for the query protein. If you wanted to know how well your method does in function prediction, I introduced, for this purpose, I introduced this evaluation measure. So you pick, for example, one threshold. This means you reduce your prediction to these 2 terms here. You calculate the so-called recall and precision for this threshold, which means which reflects how much of the actual experimental annotation you cover and how accurate your prediction is. And if you do that for various thresholds, you get a recall precision curve for one of your proteins. If you repeat this for multiple proteins, you get multiple recall precision curves. And if you want to really boil this down to one curve, you average over all the curves. And if you wanted to boil down this curve to just one number, you can take the so-called maximum F1 score, which is basically which calculates an average for every recall precision curve, for every recall precision point, and then simply takes the maximum of all these values. And so as I said, so the students implemented 2 variants. This is what the evaluation of these variants brought us in the end. So one method has only uses 2 different scores, but the scores that it calculates are quite accurate. You have another method that covers a rather low recall region with a high precision, which means so only little of your actual of the actual true annotation is predicted, but what is predicted has quite a high accuracy. And finally, method B here covers exactly the opposite, basically. So a large area, a high recall, so a high coverage of all the true experimental terms, but with a rather low precision. Okay. Any questions about this? Yeah. No? Okay. Okay. So after the exercises were finished around 4 years ago, not coincidentally, there was something called CAFA, the Critical Assessment of Function Annotations. So again, a question. So who has heard about CASP of you? Maybe. No?
Is it the function, the contest that goes on?
Yeah. Contest about what?
Predicting the structures.
Exactly. Yeah. So in computational biology, you have hundreds of methods which all try to predict, in case of function and in terms of and in case of structure, which all try to predict more or less the same thing. But the problem is that the authors who develop the methods also usually have evaluated their own methods. And in order to really get an independent assessment without any sort of bias, you sort of need to do the evaluation yourself. And CASP has been around, I don't know, for 20 years at least, and it measures how well methods are for predicting structures. And CAFA really was the first independent assessment of how well methods do for functions. And so for the prediction of protein functions from protein sequence. And this was again shortly after the exercises. So first of all, let's again explain the problem. So going big here, so it means that we really now take all the methods that are out there that predict function, and try to compare them, and problems with the for you, if you wanted to know yourself what function, what method is best, you would run into all sorts of problems, like different data sets, different input features, target classes. So not every method used the gene ontology, not every method only used sequences. The evaluation measure that I just introduced is only one variant of many. And all the methods have different strengths and weaknesses. So some are very slow, for example, some are rather fast. And you can interpret this picture here so as in two ways. So for once, it is of course very difficult to predict a function from sequence, as you can imagine, if we don't even know how to predict structure from sequence, and also getting all these different methods together and evaluating them independently is also quite a big task. Yeah, and that's why this first Kappa experiment was held. So it was quite an impressive list of organizers. So our professor was one of them, but all the other people, they came from Oxford, MIT, Princeton, yeah. Also all from the computational biology departments. And yeah, so if you want to do such an experiment, you need to follow a certain order of steps. So first of all, you of course need to find the organizers. Finally, agree on the standard that you want to predict. In our case, I already explained it. So the Kappa also used the gene ontology. But so there are a few details to this. So this is all very it seems very simple. So there are edges, there are labels, and you have a hierarchical organization of the whole thing. But if you look into the details of it, so it becomes more complicated. So first of all, the gene ontology uses different labels for the edges. So if you go from one child term to the parent, you can find sort of what is called an is-a relation, a part-of relation, a reverse has-part relation, regulates, occurs, and so on. And depending what compositions of what order of relations you find as you go from the bottom to the root, you have different so-called path inference rules. So let's say A is connected to B and B is connected to C, then it really depends on the exact composition of labels whether you actually have a relation between A and C. And but luckily, so the gene ontology itself provides rules for how to infer all the relationships in the entire ontology. But obviously, so this makes things a little bit more complicated. Then you have different evidence codes. So evidence codes are very important in computational biology because they help you to distinguish between annotations that are predicted and annotations that come from experiments. And the ones that come from experiments are again divided into various different types. So inferred from mutant phenotype, so somebody exchanges an amino acid in a protein with another amino acid and then just measures what happens overall in the cell, and from that infers the functions. Or they look at the expression in the protein in the cell, and from that derive the function. Yeah, and so on. And finally, you have the most frequent term that you can find, the IEA term. This means inferred from electronic annotation, which basically means that anything annotated with computers. And it goes on. So each annotation can be associated with different qualifiers, the most important one being not, which means that the protein does not have this function. And of course, the gene ontology itself is an evolving entity, so there are constantly protein function terms coming and going. And you obviously have to, in the CAF experiment, you have to agree on exactly one version and what type of ontology you want to use. And finally, so the gene ontology is a consortium, which means it is composed of different subunits, and unfortunately, every subunit basically has their own annotation team. They from time to time copy from each other, but not always. So there is a sort of a delay in the whole process, so that you then have to find a way to deal with all the different annotation groups for your experiments. So in CAF itself, we then finally did the following. So we only used one type of relation. So namely the pre-computed graphs by the gene ontology. We of course only used experimental evidence codes, and not even all of them, but only really the most reliable codes that are in the database, so that we are sure that the protein really does have this function. We used all the same Go version and we used only Swissprot as the annotation source. So Swissprot is a very reliable source of protein annotations. So everything that you find in Swissprot is actually manually curated by humans. And just to give you an idea of the overall difficulty of the problem. So in the molecular function ontology, we had almost 9,000 terms, which translates to 9,000 classes. And each protein, of course, can be associated with multiple classes. So we have a multi-label problem with almost 9,000 possible labels. The ontology has 7,000 leaves, so 7,000 most specific descriptors of function. The maximum depth, so the maximum number of jumps you can of labels you reach on a path from a leaf to the root is 11, and you have on average 6 children per parent term. Biological process ontology is even more complicated with 19,000 terms, and overall, and 8,000 leaves, and is a little bit more has a little bit more children per parent term, and is a little bit deeper. Okay, so now you know what to predict. Now you need, you want to know what to assign to the proteins. Now you also need, of course, the proteins that so far have no annotation. And this brings us to the following problem. So from each kappa, from each group, from each method that competes in kappa, we would like to have predictions of proteins that are not experimentally annotated right now, will be in about 4 months. So 4 months was the time period from the submission deadline to the final evaluation meeting. And we need a high enough number of proteins in order for our results to be statistically significant. And one task for you here. I'll give you 5 minutes. So how would you do that? So what is one way of finding those targets?
You could do the search for similar sequences and then see what those sequences do.
Wait. So search for similar sequences. So what is that you search with?
The sequence of this protein that we are studying.
Yeah, so but that is exactly the problem, right? So we want sequences that are not annotated right now, but that are annotated in about 4 months from now.
Okay. So we basically know nothing about it.
No, that's exactly what the problem is. Yeah. So sequences that are not annotated right now, and so these sequences will be predicted by all the competitors. They cannot cheat because they're not annotated right now, right? So they have to predict them. But in 4 months, these proteins should have annotations because we want to measure how well the predictions were. And the question now is, how do you find the proteins that are definitely annotated in 4 months from now?
What is the information do we have about these proteins?
Only the sequence. No, so yeah, I mean, we are reaching around 100 million proteins, and 99.9% of them are not annotated. So it is very easy to find proteins that are not annotated. So how did CASP do it? Do you know? No? Okay, so CASP, for example, talked to experimentalists that had just found a new structure of a protein and asked them not to publish it. Okay, so and asked to all the competitors to predict the structure of exactly that protein. And of course, in the end, so once you had the predictions, you could easily compare those to the experimental structure. But for functions, it is way more complicated because you have many different functions, and you would have to, I don't know, ask hundreds of labs in order to get something like that. And this is not feasible in this case. Okay, so I will resolve it. So it was quite a brute force approach, to be honest. So what the organizers did is they took a few model organisms of which are supposedly most interesting to study, and they asked every competitor to predict all the proteins in these organisms that are not annotated so far. And then 4 months pass by, and some of these proteins will be annotated in these 4 months. And then you simply use the few proteins that have been annotated in this time period for your overall assessment. And yeah, just to give you an idea, so all the competitors were supposed to predict 50,000 protein sequences altogether, and in the end we had around 800 of them in the end got some annotations. And of course, so as I mentioned, so SwissProt is not the only database that has Go annotations. So there are also so-called Go-A project and gene ontology itself. So in order to really make sure that none of the proteins at submission time had any annotations, you only could take those annotations which really did not exist in any of the other databases. All right. So a few statistics of these targets. of these proteins that all the competitors were evaluated on. So first of all, the organism, so human was quite overrepresented, yeast, also mouse. The evidence codes, so the experiments that were used for the annotation, were often, as I explained, inferred from mutant phenotype. So somebody exchanged amino acids and measured what happened, but also inferred from physical interaction. So they inferred the function of the protein from the proteins this protein interacts with, and also generally inferred from diode assay, which is a rather generic term for laboratory experiments. And the distribution of the targets was rather, so the distribution of the codes was rather similar to the distribution that you also find in the entire database, which is also a good sign rather. Then, as I explained, so the leaf terms really describe the most specific functions a protein can have. Most of the targets only were annotated with one function, which is not a natural distribution. So most proteins in nature probably have multiple functions, but so because of the lack of annotations, we only have most proteins with one function, and again this distribution was rather similar between the whole database and the kappa targets. Finally, what were the functions? So what functions were most common among the targets? In the molecular function homology, it was protein binding, because virtually every protein binds something in order to carry out its function, and very often also proteins bind to themselves, which is then called the so-called homodimerization activity. And in the BPO, you have processes. So processes are much more equally distributed. Cell adhesion, so cells, so the function, the process of cells sticking to each other, as one of the most frequent ones. Finally, last but not least, a very important statistic which tells you something about the difficulty of the predictions. This the so-called sequence identity to already annotated proteins. So, of course, it is very intuitive that the more similar a protein is to a protein that is already annotated, the easier the prediction becomes. So if the two proteins are not similar in sequence at all, then annotating it functionally will be much more difficult if they have, 99% of all amino acids in common. And the distribution that you see here is actually quite a good one. So it is, except for this part here on the right, it really reflects how similar the average protein that is not annotated is to the ones that are already annotated. And the bias that you see here on the right is actually some sort of human bias. So experimentalists actually prefer to study proteins that are similar to proteins that we already know something about. This is why you have this peak here on the right in both sets. Okay, so yeah, now you have now you know what you want to predict. You know the proteins that you want to annotate. You need a measure. Luckily, this measure is exactly the same that I explained earlier. So this was also another coincidence. So we talked to the organizers during the exercises and then adjusted our measures accordingly. And finally, you have everything at hand to evaluate your predictions. So here are a few statistics of what other labs beside ours used for the predictions. So almost everybody used used sequence alignment. So exactly the same thing that we did. Many of them also used machine learning based methods, so we did not use machine learning. Orthologs, which is also very similar to sequence alignment, so-called sequence profile alignment. So a sequence profile you should have heard about, or at least you will be hearing about very soon. And also literature. So literature means that so we made sure that no protein was annotated in other databases so far, but this does not mean that there is not some paper which already describes something of what the protein is doing. So by automatically mining the literature for the proteins that we want to predict, we might find something which gives us clues about its functions. Finally, also protein interactions, again, as I said, so the proteins that our targets are interacting with give us clues what it does. And same for gene expression and all the other features. A few preliminary rules for the cover. So every group was only allowed to submit one set of predictions for the targets. There was one recall precision curve for each ontology, and we did not evaluate the cellular component ontology. No measure is perfect, which means that even if a method somehow excels in one category, this means if you would look at the predictions in a different light, you may find that the order of the methods actually changes. Of course, the implementation of the evaluation code was a very sensitive subject, and this was done in a joint effort. The identities of the groups were hidden because a lot of research funding depends on the results in this experiment, and if you do bad, you may lower your chances of getting new funding. And finally, a few baseline predictors, which were sort of some sanity check for methods. One is priors, which simply assigns the same prediction to all the targets and gives each Go term as a score the prevalence of this Go term in the entire SwissPro database. So if every protein in the entire SwissPro database let's say 80% of all the proteins in the SwissPro database are annotated with the same Go term, then this Go term is assigned a score of 0.8. And finally, BLAST, basically one nearest neighbor method that simply assigns to a new query the functions of the most similar hit in the entire database. And yeah, those are the recall precision curves of the entire of all the methods in the molecular function ontology. As I said, so only one method was allowed to be submitted per group, and we chose method A, which means we landed here rather in the middle of things. But for the biological process ontology, we performed rather well. And also, if you then, for example, look at some subset of targets, in this case, the most difficult targets according to the sequence identity to the proteins that are already annotated, we actually belong to the top methods. And if you consider that students implemented this in just around 6 weeks, 6 to 8 weeks, and you were competing against the entire world, this is quite a big success. And yeah, and this is the same thing then again for the maximum F1 score. So the whole curve boiled down to 1 point essentially. And yeah, some summary of the whole experiment. So the top 3 methods called Jones, UCL, ORCID2, and Panzer. So this came from London, Italy, I believe, and Sweden. And what is striking here is that they all use sequences for their prediction. And also a method called gostruct, which is this curious pointer up here, also was entirely sequence-based, and John's UCL, so arguably the best method, said it best when with the following. So they found that the high throughput data integration based predictions contributed least to the final predictions, which basically means that in everything except for sequences, you have an extremely large amount of noise in your data, so that really only the sequences carry most of the signal that you can use for functional prediction. Which also means that we were that we were already on the right track in the exercises. Okay, let's see how much time. So we were quite glad after Kappa, but we still saw a lot of room for improvement, and this was now done not by students but by myself. So for the Kappa experiments, we again had three methods, which I will call student A to student C now. They were based on Cyblas, or a very fast similarity search. There were several problems with this method. So only one of the three classifiers was evaluated. This was a problem because we later found out that other classifiers would have actually been even better. All the three parameters that the classifiers used were not optimized at all, or even or only in a suboptimal way. Also, we would have ideally wanted to combine all the predictions in one meta-classifier. So meta-classifier means this is usually some sort of machine learning tool that you put on top of baseline classifiers, and which should almost in all cases perform better if the space classifiers have some sort of diversity and are not totally correlated. One of the methods actually had quite a severe bug, which really was detrimental to performance, and we also did not have the original implementations of the evaluation measures, which can make a small but decisive difference in performance. And also the exact evidence codes for the development of our methods. So the exact evidence codes that were used finally by the Kappa people were not known at the point where we developed the methods. And so it is quite striking also that the evaluation that we did ourselves was much better than the one in Kappa, and this is really because of this list of issues right here. Right. So what can we do in order to improve things? So first of all, you need some sort of independent test set that you can play around with without really cheating for the prediction of the Kappa targets. So what we did then is we basically simulated a so-called a Kappa 0. So we said what would have happened if the exact same Kappa experiments happened 1 year earlier. We reconstructed the exact same data sets with the exact same procedure that Kappa did. So in numbers, this means that we were allowed to use all the templates that were annotated before 2010 in order to make our predictions. The targets, so the proteins that we wanted to predict were all the proteins that gained annotations in the year 2010. Basically, this was our training set, basically. Training setup where we could optimize our methods, and finally we then used this setup here, which is exactly the Kappa setup, in order to get independent. So basically, a repetition of the Kappa experiments in order to see how well we would have performed had we done all the corrections earlier. And again, to put it into words, so from now on, the training templates will be referred to as this one, so 2010. The test set as the same thing without the smaller than sign, and analogously, everything that is used for testing then corresponds to the original Kappa targets. So what do we do with the methods themselves? So we introduced 3 parameters. We analyzed which parameters they actually had. So K, so how many of the top hits do you actually consider for your prediction? What e-value thresholds do you use? So e-value means the chance that you find such a hit by chance, actually. So you can also, instead of the K, you can also define a cutoff that is only entirely based on the e-value, and all the other algorithmic alternatives as long as they were given. So some groups, for example, experimented with different kinds of scorings, or used the sequence identity of the positive identity. And finally, you had so 36, 54, 72 parameter combinations for the different methods. And as I said, we had a training set, we had a test set, and we used our training setup in order to try out all the parameter combinations in a rather brute force way, and we simply picked the parameter combination that resulted in the best accuracy, and the resulting optimized methods are then called student A prime to C prime. Okay, so you have now your optimized baseline classifiers. Now you need to somehow combine them in the meta prediction that I mentioned earlier, and this is what we did for this purpose. So staying with the overall scheme, we tried to use the most simple machine learning tool that we could think of, which was the linear regression. Linear regression means that you have this combination of scores where the A prime, B prime, C prime indicates the prediction of one coterm for one protein by all the methods, and X, Y, C are the weights that have to be optimized by the linear regression. And finally, if you have done that, then and you want to predict a new protein, you predict this protein with all the methods, you collect all the terms that the methods predict, and you put them into this formula. So each and every term, you put it into this formula. So in case a term is not predicted by all the methods, it simply gets a probability of 0, so that you have 3 numbers really for all the terms that you predict, and in the end you ideally get a probability that reflects your trust in the prediction. One difficulty with the setup that I described was that, so if you use one data set to optimize the baseline classifier, you can no longer use it to really learn the predictions, and to learn the meta classifiers, so the linear regression. So what you do is you split the set into 2 parts called 2000A and 2010A, 2010B, and you use one of the sets to optimize your baseline classifiers. Then you use with the baseline classifiers, you can then independently predict the set 2010B. You get scores for all the coterms, and you can learn the linear regression with the scores, and then you repeat exactly with reverse rules. So first you optimize your Bayesian classifiers with the set 2010b, then linear regression with 2010a, and you simply average over the weights over these weights here that you get, the x, y, z. All right, so this is it. So this was enough for us to train the methods and repeat the Kafka experiment, and this is then what we got out in the end. So you don't have to look at the details of the differences between all the methods, but what is striking here is that the A prime, B prime, C prime meta. So this is the final best answer by us. This one you can find here for example, and as you see, it really greatly improves over other methods that you find down here. But also the Bayesian classifiers perform quite well in rather low recall regions. The one thing another reason why you want to really choose this region here, that you want to choose a classifier that performs good in this region, is that the maximum F1 score that we were using in order to really compare the methods, all the methods that were competing in Kafka by one number, this F1 score is best in regions around 0.5 recall. So it is virtually impossible to get a good max F1 score by only performing good in this area right here. So you really want to optimize for this intermediate area right here. And similarly, so this is the BPO ontology, this is the MFO ontology. Again, meta predictor greatly improved over our submission. And yeah, and this was in fact so if we had this curve at Kafka submission, so this is basically state of the art. I believe it is 1 percentage point worse than the best performing method in Kafka. Now let's not go into the details of that. And this is basically then another way you can look at the results without the curves and with different measures. So not only recall precision curves, but a couple of other ones. And you see again that meta performs pretty well, but that the ranking of the methods really depends on the way you're looking at the predictions. And finally, summarizing Kafka, or the optimizations that came afterwards. So a loose coupling, so the linear regression of very simple nearest neighbor-based methods works extremely well for protein function prediction, better than using high throughput experiments like gene expression or protein interaction. You need multiple measures really to get the full picture. There is no single method that is best in all categories, so this was not even the case for us. So some baseline predictors performed well in recall regions that we did not, so that the meta predictor did not perform well. There is still a long road ahead for the prediction overall, as you can see here. So there is a lot of room for improvement in this direction. And the problems of homology persist, like for example the domain problem, or the problem that even single amino acid substitutions can change functions. All right. Yeah, let's do one more topic. Kafka 2. Kafka 2 was held. Yeah, the meeting of Kafka 2, the assessment meeting, was this year actually. So there were 4 years in between. No, 3 years between Kafka 1 and Kafka 2. The reason was ironically that Kappa-1 was a big success. So many new methods were submitted to Kappa-1, which had to be published. We had to think of a few new ways to do the evaluation. And in the end, so Kappa-2 had, as I said, a little bit more rules. Had new prediction disciplines, which I will describe in a moment, and of course more targets. Rules. So in the first Kappa meeting, so the gene ontology was not involved. So they were quite they were sitting in the background basically without knowing about it, but they were informed for the second meeting and actually wanted to contribute to this entire process. And for that, they introduced new features in the ontology themselves. So they had now so-called annotation blacklists, which you can use in your automatic prediction method to avoid protein GoTerm annotations that really do not make any sense. So, for example, if you have one query, you align it against the entire annotated part of SwissProt. You find a very good hit, but the one function that this hit has depends on only one amino acid, and exactly this amino acid is different in your query. And of course, in this case, you should really not predict your query to have the same functions as your template. Also, if your target, for example, comes from human, it does not really make sense to annotate it, for example, with photosynthesis. So what plants do. Similarly, there are so-called taxon restrictions, which are GoTerms that simply should not be used in certain taxa. So, as for example, flower development. So all they should obviously not be used in eukaryotic organisms, but only in plants. And finally, some terms were suggested not to be used entirely for automatic predictions. For example, like S-phase, which describe a time period in the mitosis, and this process is only carried out for a very short period of time. So that is not really a definite description of the protein, and also two general terms were discarded from the assessment. And if you compare this to the rules that we already introduced, so now the gene ontology really has become quite a bit of complication. So you go from, I don't know, 10 lines of code to 100 to 1,000 really to accommodate for all these different variants, different rules. And also for Kappa-2 there was a new discipline, actually two new disciplines. So now we not only want to predict proteins that are not annotated so far, but we also want to predict annotations of already annotated proteins that gain new annotations in the time period between submission and evaluation. We have a new evaluation measure, which I will not describe in detail now, but it accounts for some of the weaknesses of the old one. For example, you could predict the entire ontology for every protein and still perform quite well. So you do not. So only the user then basically has to find the threshold that really optimally cuts off the predictions. Also, every protein was treated equally. which is suboptimal. For example, if some proteins are only annotated with one term, and other proteins with multiple terms in a very detailed way. So you should really weigh the more detailed annotations more than only the superficial annotations. And also, it makes a difference whether you annotate one protein or with a very, so one function of a protein in a very detailed way and all the other functions only in a very coarse way. So you would penalize the coarse annotations and emphasize the most detailed annotations. And this new measure, which I refer to simply as semantics distances, accounts for all of that. It does not result in curves, but only in a single number for every submission. But this number should should reflect reality better than the maximum F1 score in the end. And finally, two evaluation modes again. So some methods do not predict all the targets, but only, for example, all the human targets that you also want to somehow consider those methods and not penalize them too harshly, which is why there are two modes of evaluation. Namely, one time you penalize unpredicted targets with a recall of 0, and the other time you don't. So you only evaluate on the targets that all the methods have predicted. Changes to our classifier were minimal, so we did not repeat the exercises. So there was one student who followed a machine learning approach, but so this was rather unsuccessful for various reasons. What we changed is that all methods now can only use one last parameter, which makes the method much more applicable in practice. It is now also available in Debian actually, so you can download it and run it yourself if you wanted to. And also the database. So another free parameter that we introduced was the actual annotation database that we used. Let's not go into this detail. So one curious thing that we discovered was that by using older databases, you sometimes perform better than by using newer databases, which is due to some policy changes in the databases themselves. But yeah, let's not go into the details of this. So let's jump right into the results. So for again. As for kappa 1, you now had targets. So people were supposed to predict proteins for around 50,000 sequences. Some of the sequences became annotated. This is how many of them became annotated. So 232 in MFO in the SwissProt database. Now we also looked at this at another annotation team called EBI, 238 for them, for example, and also we looked at the full at all the annotation resources together. The reason was that there were quite a few differences in the targets between these databases, and because of that also everything that I say now is still under construction. So this was published about 2 months ago, and people are still debating what the best way is to really compile the results, but for now, so the separation stands. One of the biggest reasons for the separation is the sequence identity to already annotated targets that have already explained. So if you remember, for the first kappa experiment, this distribution looked something like that, and now it looks sort of different, right? And this is sort of because of so-called fashion changes, policy changes by the annotation groups themselves. And if you look at another annotation group, Swissprot in this case, again, a totally different distribution of similarities. So Swissprot really only, in this period of the 4 months, really only looked at proteins that were already quite similar to proteins that were already annotated. And the following slides, this is, I will jump over that now. So we had much more submissions than for CAFA1, and as I said, all the results are still subject to change. But we performed top 10 here for this measure. So those are recall precision curves for Swissprot. So for the proteins that are most similar. Two proteins that are already annotated, so you can find us as here at the student 2. Not so good, surprisingly, for BPO, at least in this measure. Sorry. So this is again MFO, but for a different annotation group. So we were good in one annotation group, but not so good in the other annotation group. In BPO, generally not so good, but curiously, number 1 for Swissprot annotations in this CCO, so for subcellular localizations. And also top 5 for the EBI targets. And to really boil it down to rankings, again, preliminary results. So according to the max F1 score, among all the methods, we have these ranks. So we are 10th for the Swissprot targets, for example. But with the supposedly better measure, we perform better in almost every category. And then if you only look at all the targets, so both annotation groups combined, if you only consider methods that submitted to all the ontologies and only in semantic similarity, we are 4th out of 55. Which is, again, quite a nice result. And that's it. Thank you.